I'm really honored to be in this land of learning, and I'm very pleased to meet together with you life science through the lens of data science. So once upon a time, there were people just like you and me, and then came the data wizard and turned out into bytes. So talking about magic, this data wizard locked us or locked our lives or data lives in a computer memory. And now we, as statisticians, the angels of big data, and you will see why I use this wording, are trying to rescue humanity by making some sense out of this data using data science. And this is why the title of my presentation is Data Science for Life, or better, Data Science for Life Science. So let me first clarify what is life science. Well, it is one of the two major branches of natural science, the other being physical science, which is concerned with non-living matters. So life science involves the scientific study of life, which is a very broad concept. And today I will focus on a few of topics related to life science, specifically epidemics, ecosystems, and then I will zoom in into human beings and talk about brain data, protein folding, and gene expression data. So this is life science. The other building block of my talk is data science. So what is data science? Well, I presume most of you know what I'm talking about, but I would like to explain my understanding of data science and why I talk about the angels of big data through a song. So if everything goes well, you should, yes, hear a video. So this song was composed by a friend under my guidance. And the chorus is the chorus of Università della Svizzera italiana. And the video is made by a schoolmate of mine. So these are all sources of big data. While someone is spying on my life, data science, data analysis, data mining. A lot of scientists are working on me. Computational astronomy, computational sentiments. Some people want to mix me with the world, and I only want to be myself. But the angels of big data are looking for a brand new world with more security and with no stress. Energy efficiency, crisis management, traffic control. Smart cities waiting for us. A single prayer was working on our data ocean. So I believe there are many statistical sailors in this room, or angels of big data. So the data continues, the song continues with other instances of data generating. And you can see also some of the fears that data science brings. Because there are opportunities and fears together. Okay, so what is data science? Well, it's a new science, relatively new now, that underpins the entire big data value chain and requires multidisciplinary skills. So it starts by trying to understand the process for for example, social interactions, evolutionary process, communication, process that underlie the generation of data. And then it deals with storing, cleaning, organizing data in databases and visualizing them. This is mainly a task of computer scientists and people in informatics. And data from different databases are enriched with metadata and are manipulated and harmonized to ensure access and possibility of perform cross queries. Statistics, which is what I do, and machine learning are at the bottom of the cycle and include the analysis of data, learning from data through models to extract information, and information has to be transformed into knowledge. And knowledge supports decision and actions that are implemented, and the consequences of our actions and decisions must be evaluated. And evaluation in turn generates new data, and the circle process starts over again. And each of the stages of this process has difficulties that arise from the fact that this data is big, or better, complex, not just big. And this poses new challenges. So people talk about smart data, but data is not inherently smart. It is us, the engines of big data, that create smart data. So we start from raw data that are processed to harmonize them, as we said. Then information is extracted, presented, and visualized. Visualization is a very helpful tool because our mind can typically process 3 information at a time. And those of you who know the card game Set know this well. Have you ever played Set? Wonderful game. So from information then to knowledge, and knowledge has to be actionable, so support decisions and actions. So those are pictures I made myself while preparing cooking with my mom. So from personalized medicine to finance, data sets are really characterized by a large number of features. And the availability of this high dimensional data set poses really new challenges to statisticians that are asked to devise techniques to extract meaningful information from this data. But fortunately, often the data contained in n-dimensional embedding can be described by a handful of variables. So a subset of the original variables, or combinations not necessarily linear of those variables, can compress the data in meaningful ways without loss of information. So in other words, one can effectively map the features of a data set onto a space of a much lower dimension. Typically, this lower dimension is called manifold, and typically is a nonlinear manifold. So specifically, let's look at this data set. Well, this is collected in 3D. And here you see 2 different angles. And the question is, can you guess the data generating process? And what is the intrinsic dimensionality of this data? Well, we do see points in 3 dimension, and we do see some structure in this data. So let's look at this data more closely. So if we rotate the image, we realize that the 3 coordinates are not free to move. There is a relationship among them. And this forces the data points to live in a manifold of a smaller dimension, specifically of dimension 2. Okay? So this 2 is called the intrinsic dimension of the data. So similarly, in this picture that is related to a different data set, the points are constrained to live in a 2-dimensional surface despite being collected in a 3D space. So again, the dimensionality of the embedding space is 3, but all points are constrained to live on a 2-dimensional plane. Sometimes, some of the points really genuinely live in 3D. Sometimes are constrained in 2D, or some subset of the points are constrained on a line, for example. So sometimes not all data points are constrained in a smaller dimensional space, but there are subsets of the data points that live in different dimensional manifold. So specifically here, we have 5,000 data points collected originally in capital D dimension 9. But the first 1,000 points are from a Gaussian distribution in dimension 1, the second 1,000 from a Gaussian distribution in dimension 2, and then 4, 5, and all the way to 9. So here you see a 3D projection of this data set. So we want a methodology that is able to tell how many submanifolds there are, assign points to the proper submanifold, and identify the intrinsic dimension of the data. To me, this is a pre-processing step of any statistical analysis. This is why I'm putting so much emphasis on this intrinsic dimension. So by using this, by leveraging on this intrinsic dimension, we have developed a Bayesian methodology that allows to incorporate expert prior opinion that might be available on the structure of the data, allows us to estimate the number of manifolds, 5 in the previous data set, 5 submanifolds, estimate the intrinsic dimension of each point and therefore of each submanifold, and quantify the uncertainty of these estimators. And this allows us to perform dimensionality reduction and to do also nonlinear clustering, because ultimately we also identify clusters of data. So to give you an example, suppose we have 4 people in our sample, and on each person we measure 3 quantities, namely body temperature, heart rate, and blood pressure. So if the first 2 person, in this case, so capital N, number of data points is 5, not big data, and capital D is 3. So we have 3 variables collected on this data set. Now, if the first 2 person are really only dead bodies, then the body temperature is around 24, 25 degrees, no heart rate, no blood pressure. So their measurements are constrained to live in a manifold with no degrees of freedom of dimension 0. They all have their measurements pretty much equal, except for some variability in body temperature. On the other hand, suppose the third person is a healthy person. So it has his or her own body temperature. And we know that there is a relationship between heart rate and blood pressure that regulates our functionality in our body. So this relationship between those 2 variables makes the intrinsic dimension of a healthy person go down to 2 because of this possibly nonlinear relationship between those 2 variables that are not free to move. There's a relationship between them. If a person, on the other hand, has some pathology like hypertension, this mechanism that relates heart rate and blood pressure can go wrong, and the intrinsic dimension can be higher than for a healthy person. So this is just a made-up example, but to give you an idea of what is this intrinsic dimension and how the intrinsic dimension can allow you to cluster person. So how does this methodology work? Well, for every person in your data set, you look at the closest nearest neighbor according to the measurement after having defined a distance measure. And then you find the second closest nearest neighbor, and you look at the distance between the measurement of the first person, which is a point in a 3-dimensional space. I'm working with capital D equal to 3 because I can visualize things, but in general, we'll be talking about capital D being much larger. So we look at the ratio of distances between the second nearest neighbor to the first nearest neighbor for each one of the person in our database. So this is person 1. The closest person might be person 2. Those are, remember, the 2 really dead bodies. Person 4, on the other hand, has hypertension. The closest person is another person with the same pathologies, and so on. So for each person, we look at the first and second nearest neighbor, and we come up with this estimator of the intrinsic dimension, which is called the 2 nearest neighbor estimator. So let's look at this methodology applies to a very well-known data set, which is the iris data set. So this consists of 50 flowers on which we measure 4 characteristics. And in the data set, there are 3 types of iris flowers: the versicolor, the setosa, and the virginica. So on this, on each flower, we measure the petal length and width and the sepal length and width. So we have a 4-dimensional embedding space. So for each of the 50 flowers, we have 5 measurements. So we apply our methodology, and we identify fairly clearly 3 clusters that are almost coincident with the 3 flower species. And it turns out that the setosa flower have their measurements that are really free to vary. There's no constraint between sepal length and petal length and width. On the other hand, the virginica flower has only 3 degrees of freedom. So the variability of those measures in this type of flower is constrained. And finally, the versicolor is sort of in between. So in this plot, you see the estimated intrinsic dimension within each group. And notice that we didn't use the group variable to find the intrinsic dimension. We only used the 4 measurements. So sometimes data, actually most of the time, data are collected with noise. So for example, here again, in a 3-dimensional space, data live on a 1-dimensional line. So the intrinsic dimension is 1, but there is noise. So noise may affect our analysis, because if we look at this data set too closely, it seems that the intrinsic dimension is 3. If we zoom out a little, we might get that the intrinsic dimension is now 2. And if we zoom out sufficiently far, we can get the correct intrinsic dimension of this data set, which is 1. So observing the data at different scale can produce different insight regarding the dimensionality of the data set. So we need to find the proper scale to estimate the correct intrinsic dimension. And we do have a way of doing this, because we go from the 2 nearest neighbor estimator to the GRIDE estimator, which stands for generalized ratio of intrinsic dimension estimator. So instead of looking at second to first nearest neighbor distances, ratios, we look at n2 to n1 nearest neighbor distances, and we let n2 increase. This letting n2 increase is like zooming out of the data. And typically we take n1 to be n2 divided by 2. So we look at ratio of 4th to 2nd nearest neighbor, 8th to 4th, 10th to 5th, and so on. And we let n2 increase. And for each value of n2, we estimate the intrinsic dimension. We get plots of this form. So the intrinsic dimension goes down until it reaches a plateau. And then if you zoom out too much, everything looks like a point that has intrinsic dimension 0. So this is it for introducing this methodology for estimating the intrinsic dimension of the data. So now let's look at some applications. So we've looked at, with the lens of this intrinsic dimension, data on protein configurations, brain imaging data, gene expression data, and COVID-19 data. And each time, a simple topological feature as the intrinsic dimension allows us to uncover a rich data structure. We also applied this methodology to data sets outside life science. For example, financial data, sport data, the outcome of MCMC simulation, deep neural network data, and social network data with different communities. So in particular, the basketball data is a joint work with a team from QUT, Center for Data Science. And in particular, here's a nice visualization of how the intrinsic dimension changes as basketball teams play an action. So this is courtesy to Edgar. Thank you, Edgar. So if you want to know more about this, you should come to the seminar tomorrow. So let's now go back to life science. And let's consider this data set that looks at folded versus unfolded configuration in a protein, and specifically is a Villen head piece. So a protein undergoes a molecular dynamic trajectory, and during this dynamics goes from a folded to an unfolded state. And the data comes from a paper that was published in Science that has the longest available simulated trajectory of this protein that perform approximately 10 transitions between folded and unfolded state in this simulation. So we expect to find different dimensions in the folded and unfolded state because those two states are metastable. So they would be considered as different attractors in the language of dynamic system. So let me try to explain what that means. Well, those states are characterized by different chemical and physical features. The folded state, as the word says, is compact and dry in its core, while the unfolded state is swollen and, in a sense, wet, with most of the proteins on the outside interacting with water molecules. So from this data, from this paper, we extract the values of 32 key observables. Namely, those are the backbone dihedral angles that the molecules in the protein form. So capital D here is 32. So we go from 3 in the previous toy examples to 32. And capital N is 31,000 approximately states in the trajectory of the molecular dynamic. So using our methodology, we indeed find 4 manifolds, 3 of them of low dimension, 11.8, 12.9, and 13.2. And 1 of the manifold is in high dimension, 23 approximately. So to test whether these distinct manifolds that we found with our approach do have some interesting interpretation, we relate them to the separation between the folded and the unfolded states that can be measured by the fraction of native contacts. So there's a quantity Q, which can be easily estimated from the configuration of the system. And if Q is close to 1, then the configuration is folded, while if it's close to 0, then the protein is in the unfolded state. And we find that the vast majority of the folded configuration, when Q is bigger than 0.8, are assigned to the high dimensional manifold, while the unfolded configuration, when Q is less than or equal to 0.8, are most of the time assigned to one of the low dimensional manifold. So this implies that a configuration belonging to the low dimensional manifold is almost surely unfolded, and it has less degrees of freedom. So this was very interesting because, as I said at the beginning, a geometric feature uncovers an interesting structure in the data. We also analyzed a set of time series from functional resonance imaging, fMRI, of the brain. So this time series represent both the blood oxygen level development, the bold signal of each of approximately 30,000 voxels that capture the activity of a small portion of the brain. So we consider a single subject fMRI recording containing 202 images that were collected while the subject was performing a visual motor task. So in this case, capital N, number of data points, is 30,000, and capital D is 202. So we're increasing capital D. Now, we apply our methodology and we find that the task relevant voxels are almost always assigned to the manifold with high dimensionality, while the low dimensional manifold mostly includes noisy voxels. That is, voxel where the corresponding time series is dominated by low dimensional noise. So let me show you this with an image. Here, the red voxels are those assigned to the high dimensional manifold, The blue voxels are the task-relevant voxels, and the green voxels satisfy both criteria. So are the task-relevant voxels that are assigned to the high-dimensional manifold. Now, because in those images you see very little blue, it means that all the task-relevant voxels, or most of them, the blue ones, are assigned to the high-dimensional manifold, meaning that the intersection, which is the green area, is what you see rather than seeing the blue dots. And moreover, those task-relevant voxels are concentrated in the occipital, temporal, and parietal cortex. And neuroscientists found this quite interesting, and will try this methodology on other type of data, other fMRI time series where the subject is performing different tasks. So let's move to gene expression data. So genes are differentially expressed in patients when compared to controls. And identifying those genes that are differentially expressed is of great importance for diagnosis, to identify therapeutic targets, and to understand the underlying mechanism driving this pathogenesis. Again, the intrinsic dimension is a methodology that allows us to identify those differentially expressed genes, and at the same time to quantify the uncertainty of the resulting nonlinear clustering of patients. So the different manifolds here cluster cases and controls. And knowing those clusters and which genes are differentially expressed is helping in developing personalized treatments and advancing precision medicine. So let me show you the specific data we have been working on. So we have 69 tissues colored by cell topology that you see in this image. The vertical line divides cases and control groups. So on the x-axis, you first see the 38 cases that are clustered in the low dimensional manifold. And note the huge dimensionality reduction. Here, capital D was close to 17,000, and the intrinsic dimension is down to 7. So while cases end up in the low dimensional manifold, most of the controls end up in the higher dimensional manifold, namely their intrinsic dimension is approximately 13. But there are some controls, the gray and the red ones, the bottom right part of the picture, that belong to the low dimensional manifold. Now there's a color coding that is beyond the purpose of this talk, but the genetists were interested and found an interesting explanation of why the gray and red control types ended in the low dimensional manifold. So what is the map that projects this 17-dimensional data set in a space of just 7 dimensions? Well, this is an open question and we are working on it. But again, genetists are very interested in finding this map, this projection, which is typically a nonlinear projection, because they believe this might encode what they call pathways. So pathways that are also known as gene regulatory network can turn genes on and off. And those are a collection of molecular regulators that interact with each other and will determine the functionality of a cell. So this methodology that I've been telling you about and I've been using in these examples is fairly easy to use. There is an R package that was created by a former PhD student of mine, now assistant professor at Cattolica University in Italy. And there's also Python code developed by the colleagues in Trieste in Italy. And this research line was developed in a few papers. Here you see a reference series. And most of the papers, 4 of them, with the methodology developed, were published in scientific reports 2017, 2020, 22, and 2023. Plus this Journal of Statistical Software or Intrinsic Dimension. So this is the methodology. Plus we have the specific applications that were published, for example, the NBA basketball data in the Annals of Applied Statistics. So I'm happy to take questions, but I'm not done yet. I will get back to this intrinsic dimension methodology, but before that, I want to talk about another interesting application, which is another joint work with QUT, Center for Data Science, and specifically Julie Vercelloni and Kerry Mengersen. Julie was supposed to be here and tell you about the Great Barrier Reef biodiversity study that we have run. Unfortunately, she couldn't make, but she helped a lot preparing this part of the slides together with Luisa Piancastelli. She's a PhD student with Niall Friel at University College Dublin. So again, this is a joint collaboration across the globe. So the Great Barrier Reef, as you probably know better than me, is 2,000 kilometers long, almost the size of Italy, and is the home of approximately 600 types of corals and other organisms. It is the world's biggest single structure made by living organism, the reef builder's coral, also known as hard corals. And it's so big that you can see it from the space. So the Great Barrier Reef is a very fragile ecosystem facing various impacts related to climate change, such as coral bleaching. And this happens when the water is too hot for too long. So under heat stress, the coral expel their symbiotic algae and their white skeleton becomes apparent. And at this stage, corals are not dead yet and can get new algae from the water column. However, if the heat is too long, corals are too weak to get another algae and die. And once they die, they become brown in color, as you see in the image here. So bleaching is the biggest threat to the reef because it impacts across broad spatial scales. And next summer will be critical for the reef as another bleaching event may occur associated with the El Niño cycle, which is already impacting the southeast part of the Pacific, and the hot water is now moving towards Australia. Another threat to the reef are cyclones. So while cyclones are a natural phenomenon, like a fire bush in the forest, their intensities are predicted to increase with climate change. So it means that they will impact a larger part of the reef. And it is forecasted that approximately between 70 and 80% of the reef will suffer worldwide due to an increase of 1.5 degree temperature over the next 25 years. Now, the latest output from the Intergovernmental Panel on Climate Change demonstrate that this agreement of 1.5 degree increase won't be met anymore. And we are indeed already tracking an increase of 2 degrees temperature, meaning that more coral reefs will suffer. So there is an emergency for coral reef research. And in order to better understand this data, we put together this cross-border team. So the study starts with data on benthic composition of 58 reefs that are located across the barrier during and after disturbances. So the data are part of a big sea view survey that use machine learning algorithms to automatically extract coverage of benthic communities from geotagged images. So this is an example of citizen science that was developed under a project called Virtual Reef Diver. So as part of the sampling design, we observe data at different locations, so 58 locations. And for each location, we have a vector of composition of the reef coverage. And because this is composition, we have percentages or proportions that sum up to 1. And the goal is to estimate the relative coverage of benthing groups by properly accommodating for the nature of the data. So we want to identify group of reef with similar composition and investigate how disturbances impact on clustering. So here you see the data that were collected across 23 reefs in different transects from 2012 until 2017. So we have 4 years of data. And this time series is really unique because during these times, a series of disturbances impacted the north section of the reef. In 2014, we have the cyclone Ita, or Ita, I don't know how you spell it, how you say it. I would say Ita, being Italian, that hit the reef at category 5. And 1 year later, the cyclone Nathan hit again with category 4. In 2016, there was a match bleaching event that impacted the entire northern part of the reef. And another bleaching event happened in 2017. So for each reef location, we know whether or not they were impacted by these events. And we know we collect data on their compositions. And we study how this composition changes over time. So here you see the heat map that is the outcome of our analysis for 2012. So blue cells show pair of reef locations with high chance of belonging to the same cluster. So you see 4 patches of blue, dark blue colors, meaning that in 2012, before disturbances, the model estimated 4 clusters. So the main cluster was composed of 40% hard corals and a similar amount of algae and sand and 20% of soft corals. Then we moved to the subsequent years, 2014, 2016, and 2017. And during and after these years, we had these disturbances. So we want to see how the clustering changes. And here you already see from this heat map over the years that instead of 4 well-distinct clusters, in here 2014, there's probably 1 cluster only, and then 2 clusters in 2016 and 2017. So to understand the extent to which the cluster have changed, remember that there were 2 tropical cyclones and 2 bleaching events during 2014 and 2017. And the reef composition has changed, going from 4 clusters in 2012, a single cluster in 2014, and 2 clusters in 16 and 17. So the main cluster, composed of algae, hard coral, remained fairly stable over time, but we lost the clusters dominated by soft colors in yellow, that was only visible in 2012, and the sand and hard color cluster in orange. So the analysis shows that there is a decreased number of clusters over time in comparison to 2012. And there is an increased within group similarity across the entire Great Barrier Reef in response to disturbances in the northern section. So despite the fact that disturbances happen in the northern section, the effect has propagated throughout the Great Barrier Reef. So overall, there is a reef homogenization of benthic composition, and this, in a sense, means less biodiversity. So we plan to continue these studies with additional data, and we look further into these compositional changes. And as I said, our international collaboration helped to address these critical knowledge gaps by providing new analytical methods. So it was very interesting to work on this really unique data. So let me move to COVID-19. So as Angels of Big Data pretty much has worked on COVID-19 data. This is again a joint work with QUT, specifically with Abhishek Varghese, Edgar again, Kerry, and Francesco Denti. So I have to note that Abhi, when working on this data, was a really young student, completing his bachelor and starting his master. So it really did a very good job. And unfortunately, it's not here. I couldn't make it. So here we use 3 data sets to explore the disease and spreading dynamic of COVID-19 in countries all over the world. Specifically, we have the Oxford Stringency Index that describes the stringency of government measures by recording the number of government policies in each country and their strictness. So we have collected for each day, starting from May 2020 all the way to March 2021, this COVID stringency index. Then we have collected the COVID new cases and the COVID deaths per million population. So basically, we have 115 countries with less than 1 million population, so smaller countries were excluded, and as well as country with more than 20% missing values were also excluded. For the other countries, the missing values were imputed with some statistical methodology. So the number of data points is 115 countries. And as I said, for each country, we record 3 time series over the period from March 2020 till end of May 2021. So this is a period of 454 days. So capital D is 454 times 3. So over 1,300 data points. So what do we do with this data? We use the Intrinsic Dimension methodology. And the main conclusion is that high-income countries are more likely to lie on low-dimensional manifolds. So ultimately, we identify 2 manifolds. And in the low-dimensional one, we have most of the high-income countries. And why that happens? Well, this outcome is likely arising from aging populations in those high-income countries, where the average age is higher, and older people are more likely to have comorbidities, and this has most likely caused an increased per capita mortality burden from COVID-19. So here I'm showing you some images. By the way, this paper has just been accepted last week in Scientific Reports. So here you see the mean and confidence interval in the shaded area of the data. The first plot is the stringency index, then we have the new cases, and then the new deaths. And we have the time series separated out for the 2 clusters, the 2 manifold. So the 2 clusters are very clearly identified, and you see that there are different dynamics of the stringency index. Well, at first, all countries. So time 0 is the time where we observe the first 100 COVID deaths. So the time series are aligned. At first, all countries responded with strong measures, but then the green countries relaxed those measures earlier in time, and then there was the second wave. They increased them again, and so on. So, but you really see different dynamics of the 3 time series. And here you see on the left, the median posterior intrinsic dimension estimated for each country, the green and the red countries. And we clearly have 2 clusters, very well identified. Australia is in the red cluster regions together with South America, China, and some African countries. Italy, on the other hand, is in the red cluster, in the green cluster, together with most of rest of Europe, North America, Canada. And on the right, you see the posterior estimated intrinsic dimension. As I said, this is a Bayesian methodology, so we can identify the uncertainty in our estimates. But if we take the posterior mean, you can clearly see that we have 2 clusters of one with intrinsic dimension about 9, the green, and the other one with intrinsic dimension bigger than 12, or around 12. So here you see the world map of countries colored by their intrinsic dimension, the red and the green countries. And notice that in the analysis, we didn't use any location information. So this clustering happened just by using the intrinsic dimension methodology. And here you see the distribution of countries by income in each manifold. Left, you see the countries with high intrinsic dimension. And to the right, you see countries with lower intrinsic dimension. And the darker green is the color, the higher is the income. So countries with higher income, on average, are more likely to lie in the low dimensional manifold. So the ones on the right. And this income classification is performed by the World Bank that assigns one of the 4 income levels to each country, ranging from low to high income. So as I said, a range of factors may explain the skew distribution of income level towards the low intrinsic dimensional manifold. As I said, high income countries usually have aging population that arise from declining fertility and improving mortality due to income growth. And this also changes the health behaviors and they typically also have a higher education level. And aging population in many high-income countries have played a role in creating a greater mortality burden due to COVID because of higher vulnerability of population groups over age 65. So we have performed the analysis over a different period of times of the pandemic, and we see how the cluster change through time. So this is the period end of June 2020, mid-October 2020. And I've one last application I could talk to you about, but this is related to Swiss data. But I rather stop here and take questions.